gram-positive bacilli. Now, so that it won't seem too overwhelming for us all, we're going to categorize the gram-positive bacilli. So we can actually group them into those that form spores and those that do not form spores. For the spore-forming gram-positive bacilli, we will be talking about the Bacillus species and the Clostridium species. For the non-spore-forming gram-positive bacilli, we can actually further uh, categorize them to according to whether they have regular shape and staining properties, which are the Listeria and the Erysipelothryx species, and to whether they have irregular shape and staining properties, which we further categorize to those with acid-fast staining and those that are not acid-fast staining and other bacteria. Let's start with the Bacillus bacteria. So generally, the Bacillus species are gram-positive. They are endospore forming. Most of them are motile and most of them are saprophytes, meaning they feed on dead organisms or decaying organic material. In fact, they're very versatile in degrading complex macromolecules. The primary habitat of Bacillus species is soil, and it actually can survive in the environment for many years. The bacteria have square ends and they are arranged in long chains, and the spores are centrally located. Some of them they can be paracentral, but mostly central. On culture, it shows a cut glass appearance. Bacillus cereus show hemolysis, while the Bacillus anthracis do not show hemolysis. So Bacillus spores are resistant to environmental changes. They're able to withstand dry heat and even some chemical disinfectants for moderate periods. So we have to sterilize by autoclaving them. They are able to persist for years on dry earth because they simply need nitrogen and carbon for energy and growth. Let's talk about the Bacillus anthracis. So the Bacillus anthracis bacteria are large block-shaped rods or boxcar-like rods. They have central spores that develop under all conditions except in the living body. So the reservoir is zoonotic. So they can be found in hoofstock of sheep, cattle, and goats, in the fur and the hides of animals, and from the soil. So the portals of entry to a person's body is through injured skin. This causes cutaneous anthrax, and it is around 95% majority of the cases. The bacteria can also be inhaled. The spores of the bacteria can also be inhaled in the fur or the hides, causing inhalation anthrax. That's why it is also called wool sorter's disease, because textile workers are at high risk and they contribute up to 5% of cases. We also have gastrointestinal anthrax and injection anthrax, but these are very rare. Now let's talk about the virulence of the bacteria. A major contributor to the virulence of the bacteria is its capsule, which is made of polygamma D-glutamic acid. It is antiphagocytic, and the gene that expresses the capsule formation is carried on a plasmid. The spore also contributes to its virulence. So the spores are able to withstand extreme environments. The anthrax bacterium secretes an exotoxin called the anthrax toxin, which is made up of 3 proteins. One of the proteins is protective antigen, which binds to specific receptors and forms a membrane channel to allow entry of the 2 other factors, edema factor and lethal factor, into the cell. So edema factor, along with the protective antigen, forms edema toxin. This toxin causes edema. The third factor or lethal factor, along with protective antigen, form lethal toxin. And this is a major virulence factor and the cause of death in infected animals and humans. So, how do patients with anthrax present? For cutaneous anthrax, they usually present with a pruritic papule around 1 to 7 days after entry into a break in the skin. This progresses into a vesicle and then to a necrotic ulcer. And you will notice a characteristic black scar. The patient will also feel systemic symptoms such as fever, body malaise, but the lesion eventually dries, loosens, and separates, and it actually heals. The healing is by granulation, so you would expect that there is scar formation. Antibiotics won't change the progression of the disease or the illness, but it prevents dissemination. And in 20% of patients, some go to sepsis and then progress to meningitis and eventually die. So for patients with inhalation anthrax, they can actually show symptoms even as late as 6 weeks after exposure. And the early manifestations are hemorrhagic necrosis and edema of the mediastinum, which presents with substernal pain. And on X-ray, you will see a mediastinal widening. Once the pleura is involved, the patient will have hemorrhagic pleural effusion. Of course, cough is also there, and sepsis may also happen. If the patient got inhalation anthrax, the spread is usually hematogenous, and it is very fatal. It has a high fatality rate. Like I said, gastrointestinal and injection anthrax are rare. Now, how do we diagnose a patient to have anthrax? So, we can examine the fluid or pus specimen from a local lesion, and on culture, you will see the characteristic medusa head. Okay? And compared to the other species of the same genus, Bacillus anthracis is actually non-motile. The drug of choice is ciprofloxacin, but you can also give penicillin G, doxycycline, erythromycin, or vancomycin if ciprofloxacin is not available. But more important than treatment is to prevent the disease from happening in the first place. So there is actually a vaccine. It is a live attenuated bacilli with spore suspensions or with protective antigen or the PA. Okay? And for those with possible exposure, they can take a prophylactic dose of ciprofloxacin for 60 days and 3 doses of the vaccine. There is also a new drug, raxibacumab, whose mode of action is to prevent binding of the protective antigen to host cell receptors. So this also can be used for prophylaxis. Now we talk about Bacillus cereus. Bacillus cereus infection causes food poisoning. And there are 2 forms of the infection. First is the emetic type, and the other is diarrheal. So the emetic type is usually associated with fried rice, milk, and pasta. And it presents with nausea, vomiting, and abdominal cramps. But it is self-limiting and usually goes away within 24 hours. And this happens around 1 to 5 hours after ingestion of the emetic toxin. The diarrheal type is usually associated with meat dishes and sauces. Manifestations present after an incubation of around 1 to 24 hours. And it presents with profuse diarrhea and abdominal pain and cramps. Take note that Bacillus cereus can also cause eye infections and local wound infections, and even systemic infections. The diagnosis is usually clinical and history because presence in the stool is not diagnostic because Bacillus cereus can also present in normal stool specimens. For the treatment, vancomycin or clindamycin with or without an aminoglycoside can be used for serious infections. Ciprofloxacin can also be used for wound infections. It's also important to take note that Bacillus cereus is resistant to penicillins and cephalosporins, and using these drugs will be of no use. Bacillus subtilis is actually a non-pathogenic species, and it is usually only used as a research organism. It also has commercial use such as enzyme production, used as an insect toxin and as a fungicide. Now let's talk about Clostridium. So the Clostridium bacteria are large anaerobic gram-positive motile rods that are able to decompose proteins or form toxins or both. The natural habitat is soil or sewage, or it can also be found in the gastrointestinal tract of animals and humans where they live as saprophytes. And the Clostridia are the causes of the diseases tetanus, botulism, gas gangrene, and pseudomembranous colitis, which we will be talking about one by one. So most bacteria are motile and they have peritrichous flagella, and the spores are wider than the diameter of the rods. They are anaerobes, and colonies vary among different species. For example, the perfringens produce large raised colonies, and the tetani produce smaller colonies. So these bacteria can ferment sugars or digest proteins or both. Now let's talk about Clostridium tetani. This is the bacterium that causes tetanus. It is found in soil and in the feces of animals. The spores are terminally located. So the types are distinguished by specific flagellar antigens in which all have the same O antigen, and all of them produce the same type of neurotoxin, the tetanospasmin. So tetanospasmin is the toxin produced or secreted by Clostridium tetani. It is plasmid encoded. So the toxin has two peptides. One of the peptides, the larger one, initially binds to receptors on the presynaptic membranes of neurons, and it migrates to the cell bodies of the neurons, going to the spinal cord and the brainstem, where it diffuses to terminals of inhibitory cells. There, it blocks secretion of inhibitory glycine and GABA, causing loss of inhibition to the motor neurons. So because of that, wala na yung mag-inhibit sa neurons, so there is hyperreflexia, muscle spasms, and a spastic paralysis, meaning the muscles are tense, and because of their tense and because of the tension, they are unable to move. Clostridium tetani is not invasive. The infection remains localized in the area, for example, in the wound or in the burn or even in the umbilical stump of newborns. It is the toxin that spreads and reaches the brainstem and spinal cord, causing the effects. So the illness usually manifests after an incubation period of 4 or 5 days to 3 weeks. And the disease is characterized by tonic contraction of voluntary muscles. So the muscle spasms first involve the area of the injury at first and then progress to affect the muscles of the jaw. So this is what we call lockjaw or trismus. Trismus is the medical term. It's important to take note that the patient is fully conscious during this time, so the pain is going to be very intense. Death usually happens because of interference with the breathing, and mortality rate is very high. How is it diagnosed? Diagnosis is done by clinical manifestations and a history of injury. Unfortunately, results of treatment are poor, although you can use muscle relaxants, sedation, assisted ventilation, and you can also do surgical debridement on the wound. But the emphasis now is on prevention. So prevention can be done with active immunization with toxoids and aggressive wound care. And you can also give a prophylaxis with the antitoxin. So when you give antitoxin, you're hoping to be able to neutralize toxins that have not yet been fixed to nervous tissue, and it is injected in the area of the wound. Penicillin is also given as antibiotic for the wound. Tetanus is a totally preventable disease, so that is why universal active immunization are mandatory. So toxoids are given during prenatal checkups. This also helps avoid neonatal tetanus. Also, tetanus is the T in DPT. So it is given along with the vaccines for diphtheria and pertussis. Now, botulism is caused by Clostridium botulinum. So this is also found in soil and animal feces. So the types of botulinum are distinguished by the antigenic type of toxin they produce. The spores are highly resistant to heat and they are able to withstand 100 degrees Celsius for several hours. But this is lessened with low acid or high salt concentration. Let's talk about the toxins secreted by the Clostridium botulinum. There are actually 7 antigenic varieties from A to G. So A, B, E, and F cause human illness. A, B, and F cause infant botulism. A and B are from foods. E is found in fish products. C is found in birds. And the G is not associated with disease. Now, the botulinum toxin is absorbed in the gut and then it enters the circulation. It binds to receptors of presynaptic membranes of motor neurons in the peripheral nervous system only. So when we say PNS, it includes the cranial nerves, right? The botulinum toxin is unable to cross the blood-brain barrier, so it does not affect the CNS. Specifically, the light chain or L chain of the botulinum toxin proteolyzes the target snare proteins in the neurons. So this inhibits the release of acetylcholine at the synapse. So therefore, there is no contraction. So the patient manifests with flaccid paralysis. Take note that the snare proteins affected are synaptobrevin, SNAP25, and syntaxin. So the botulinum toxin is among the most toxic substances known. In fact, if you inject a mouse with a specimen, for example, the serum of a patient, the mouse will die very fast. Here's an interesting fact. The botulinum toxin, exotoxin A, is also used commercially for wrinkle removal. Most cases are due to intoxication from ingestion of food with the bacteria. So most common are spiced or smoked or canned alkaline foods that are eaten without cooking. For infant botulism, the babies ingest the spores in household dust, but most commonly honey when they are given honey. So this results due to the absence of competitive bowel microbes in the baby. Wound botulism and inhalational botulism are very rare. Now, the patients manifest with the clinical findings or the manifestations around 18 to 24 hours after ingestion, and they present with visual disturbances due to incoordination of their eye muscles, because remember, it causes paralysis of muscles, right? So when the muscles of the eyes are paralyzed, there will not be any coordinated movement anymore. So the vision is disturbed. Also, inability to swallow and speech difficulty, because if the cranial nerves that are controlling those actions are affected, that's why they present with such. Remember, there is cranial nerve affectation, right? So if The cranial nerve that is affected is the one that contributes to respiratory function. If it is affected, it can lead to respiratory paralysis and eventually cardiac arrest. Again, there is no central nervous system affectation. So the patients remain fully awake until shortly before they die. For infant botulism, they usually start with poor feeding and weakness and paralysis. So this is what you call the floppy baby syndrome. So remember that botulism is a reportable case. Diagnosis is by detection of the toxin in serum or gastric secretions or in the stool. And you also use mouse bioassay, which is very sad. You take a serum from a suspected patient and then you give it to the mouse. And if the mouse dies, that confirms that the toxin is present. For infant botulism, you will not be able to find or detect the toxin in serum. So you can only use other secretions. Now, the treatment is only supportive, and you can assist ventilation with a mechanical ventilator for up to 8 weeks. And this actually has reduced the mortality rate from 65 to as low as 25%. The antitoxins are equine, meaning they are collected from horses. They are trivalent, but they do not reverse the paralysis. Only that if they are administered early, you can prevent the illness or the disease or the paralysis from worsening. For infant botulism, what is given is the human-derived botulinum immune globulin, which is very expensive. So actually simpler and more important to prevent this from happening in the first place. So how? Remember that canned foods or canned goods must be cooked or heated sufficiently to destroy the spores. This is done by boiling for at least 20 minutes. Now, the two Clostridia we've been talking about were actually non-invasive Clostridia. Now, let's talk about the invasive Clostridia. The first is Clostridium perfringens. So, this causes myonecrosis and gas gangrene. On blood agar, it shows double hemolysis. And on egg yolk agar, it is non-motile but with rapidly spreading growth. It has at least 4 toxins that are identified. No? And these have lethal, necrotizing, and hemolytic properties. The alpha toxin is a lecithinase, meaning it splits lecithin in cell membranes. So this causes platelet aggregation, forming thrombi in small blood vessels. Now, if these thrombi are lodged, it causes poor tissue perfusion, which causes necrosis. The theta toxin is similar to the alpha toxin with hemolytic and necrotizing effects, but it is a cholesterol-dependent cytolysine that forms pores in the cell membranes. The epsilon toxin is the one responsible for edema and hemorrhage. And the CPE, or the Clostridium perfringens enterotoxin, induces intense diarrhea. This involves marked hypersecretion in the jejunum and the ileum. This causes diarrhea and loss of fluids and electrolytes. So, the spores of Clostridium perfringens are introduced by contamination. When the spores enter the body, they will germinate and multiply and ferment carbohydrates in tissue, producing gas. This interferes with the blood supply and causes necrosis of the tissue. The infection spreads in 1 to 3 days after contamination of the wound and causes crepitation in subcutaneous tissue and muscle. It also causes foul-smelling discharge, rapidly progressing necrosis, and systemic symptoms like fever, hemolysis, toxemia, shock, and ultimately death. The diagnosis is done upon finding the presence of large gram-positive rods in specimens. The spores are not usually seen. So the treatment is immediate and extensive surgical debridement with excision of devitalized tissue. This is the most important. You also give penicillins to avoid further infection. Hyperbaric oxygen can also be used, and it actually detoxifies the patients rapidly. Can also give polyvalent antitoxin in the form of concentrated immune globulins, but this should not be relied on. Now let's talk about Clostridium difficile. Actually, now it is already called Clostridioides difficile. This causes pseudomembranous colitis and antibiotic-associated diarrhea. It's diagnosed upon detection of the toxins in the stool and by observation of pseudomembranes or abscesses by endoscopy. So patients who have diarrhea and have been taking antibiotics are at risk. So the diarrhea may be watery or bloody and associated with abdominal cramps, leukocytosis, and fever. So the offending antibiotics suppress the normal flora in the colon, allowing the bacteria to overgrow. The most common antibiotics associated with pseudomembranous colitis are ampicillin, clindamycin, and your fluoroquinolones or quinolones. Now, Clostridium difficile has two toxins. Toxin A is a potent enterotoxin, and toxin B is a potent cytotoxin. So the activity of these toxins result in apoptosis, capillary leakage, and cytokine stimulation, causing inflammation of the colon or colitis. So the treatment for this is to discontinue the offending antibiotic and to give oral metronidazole, vancomycin, or fidaxomicin. Now we're going to talk about one more group of gram-positive bacilli. For the irregular, non-spore forming bacilli, you have those that are acid-fast staining bacteria, which are the Mycobacterium. But Mycobacterium will have a different lecture of its own. So we will talk about the Corynebacterium. So the Corynebacterium generally have irregular swellings at one end, thus the club-shaped appearance. And they're also known to have the Chinese character appearance. So you will observe metachromatic granules irregularly distributed within the cell, and they can be seen to form black colonies with the brown halo on cystine tellurite agar. There are four biotypes of Corynebacterium: gravis, mitis, intermedius, and belfanti. Take note that all four of them are able to secrete the diphtheria exotoxin. Now let's talk about the toxin. So the name is Diphtheria Toxin. It is heat labile, single chain, and has three domains. The toxin usually breaks into two fragments, fragment A and fragment B. Fragment B has two domains, the receptor domain and the translocation domain. The receptor domain binds with the CD9 protein on host cell membrane and the heparin binding epidermal growth factor or HBEGF. This triggers the entry of the toxin into the host cell. The translocation domain acidifies and causes development of a protein channel. This allows the movement of fragment A into the host cell cytoplasm. Now, fragment A inactivates elongation factor 2 and then causes an abrupt arrest of protein synthesis in the cell. So, this abrupt arrest causes necrosis and the neurotoxic effects of the toxin. So, this includes demyelination, paralysis of the soft palate, and paralysis of eye muscles and even extremities. The toxin is absorbed into mucous membranes. This causes destruction of the epithelium and superficial inflammatory response. So, because of the destruction of the epithelium or necrosis, an exudate of red and white cells are formed. So, this is actually the grayish pseudomembrane over the tonsils and the throat, which actually bleeds when you try to remove them because of the tearing of capillaries. Also, the lymph nodes in the neck will enlarge, causing marked edema. So that's why patients with this present with a bull neck presentation. The toxins may actually be absorbed and may cause distant organ damage too. So they may cause fatty infiltration and necrosis in the heart muscle or myocarditis and even other organs like the liver or the kidney and nerve damage. So the diagnosis is clinical. Treatment must not be delayed for lab reports. But you can also use the modified immunoprecipitation method, but we're not going to talk about that anymore. You can also use PCR, and you can actually see, you will also find other assays in your book, but they are not widely available. So treatment for diphtheria involves rapid suppression of the toxin-producing bacteria by antibiotics. So penicillin and macrolides are used. And you can also give antitoxin. But more importantly, is to prevent the disease from happening in the first place. So you can do this with active immunization with the diphtheria toxoid, which is actually combined with the vaccines for pertussis and tetanus.